Hello, my name is Jaqueline Geraldis. I am a PhD student at Dr. Epilepsy's group, and today I'm going to present my work, which is entitled Unraveling the Epigenetic Mechanisms Involved in Mesial Temporal Lobe Epilepsy with Hippocampal Sclerosis. Mesial temporal lobe epilepsy associated with hippocampal sclerosis is one of the most frequent and most severe types of epilepsy. For these patients, a surgical procedure may be a therapeutic alternative, and tissue obtained from these surgeries can be studied. DNA methylation is one of the most important epigenetic modifications, and it is directly associated with gene expression regulation. Whole genome bis5 sequence, ADGBS, is a current gold standard approach to study DNA methylation in the entire genome, also known as methylome. ADGBS provides a complete DNA methylation profile, including different regions as intron, exons, and promoters. Since epigenetic mechanisms play a relevant role in many neurological disorders, we aim to investigate whether a different methylation profile is present in tissues obtained from patients with mesial temporal lobe epilepsy associated with hippocampal sclerosis using whole genome bis5 sequencing. DNA was extracted from the hippocampal tissue with confirmed hippocampal sclerosis using the phenochloroform protocol and quantified with qubit high-sensitive kit. We have analyzed 11 samples from patients and 4 controls from autopsy. In addition, samples from patients were divided into 2 groups. Group number 1 is composed of 5 patients with less than 20 years of disease duration, and group number 2 had 6 patients with more than 20 years of disease duration. The term year of duration comprised the time interval between diagnosing and surgery day. Next, we performed the bisulfite conversion and the preparation of the sequence library using the ACell NGS methylsec DNA library kit. Then, whole genome sequence was performed at Illumina NovaSeq 6000 platform. After sequence, data was treated with bioinformatical tools, Trim Gallery and Bismarck, and the statistic package BSEC. To analyze differently methylated regions, DMRs, Bismuth was used. For this study, we had 3 comparisons. The first and the second were patients group number 1 versus control and patients group number 2 versus control. The third was patients group 1 versus patients group 2. This is still an ongoing study, and to date we found more than 1,000 differently methylated regions when comparing the patients to control. The identification of different methylation regions is underway, as well as the correlation of the methylation profile with the transcriptome profile of the same tissue. For final conclusion, we have identified many different methylation regions in tissues from patients with mesial temporal lobe epilepsy associated with hippocampal sclerosis, suggesting that epigenetic modifications may play an important role in the underlying mechanisms leading to hippocampal sclerosis. The identification of specific methylated regions and its correlating with gene expression data may indicate new targets that could be used for therapeutic purposes in the future. I would like to thank Dr. Lisa Lopes and Dr. Daniela Doghini for the orientation, my co-authors, group and lab members for all the help and the funding sources. Thank you for your attention, and if you have some question, my email address is on the first slide.